Good evening, everyone, and I welcome you to the Biomedical Data Science and Drug Discovery program. This 4-month program has been designed to take students through a step-by-step process of a project-based biomedical data science and computational biology training, along with mentor support and guidance by industry and academic experts from all across the globe. This collaborative program has been designed for students and brought to you by Pine Biotech USA, Faba Academy India, Pharmatopus India, and Pro Biotechnica India. Here we have the team from Pine Biotech, Dr. Harpreet Kaur, Dr. Mohit Mazumdar, and Ilya Brodsky. Dr. Sujit Tangat Paliwar is a co-founder of Pharmatopus. Neetu Singh is the CEO of Pro Biotechnica. And from the Faba Academy, we have a panel of mentors by Dr. Ajit Kamath, Dr. Ramesha, Dr. Saxena, Dr. Reddy, and Dr. Kakkar. And we are all going to be meeting them throughout this 1-month period, and they will help you through the sessions and guide you through this entire training program. So the first part of this 4-month training program is the OmicsLogic Genomics for Biomedical Data Science and Drug Discovery, which is starting today and will end on May 18th. We will be combining online sessions, self-guided study materials, and practical assignments for an immersive experience that has proven to be effective in our OmicsLogic programs. I would now like to invite Ilya Brodsky, who is the CEO and co-founder at Pine Biotech, to talk more about this training program.
So great. Thank you very much, Bipsa. And I'm excited to see all of you joining today. So as Bipsa mentioned, my name is Ilya Brodsky. I have been working with the Pine Biotech team, as well as our collaborators at the Tauber Bioinformatics Research Center, for now a few years, bringing these programs to a variety of settings. And as you can see on this image, we've done workshops at universities, worked with faculty, clinicians, with students. Really, our mission and our vision is to democratize bioinformatics, meaning we want to make it much more accessible, much more easy to get into and leverage in application to a variety of different types of applications. Because what we have found is that so many times training is focused on technical applications, that's one side of the spectrum, or it is very limited in the scope. And so what we wanted to do is to expand that scope and allow participants from diverse backgrounds apply bioinformatics and big data analysis skills to a variety of challenges that they are interested in cancer, in neurodegenerative diseases, genetic diseases, infectious diseases, agriculture, and biotechnology. And I think what's important is that there is a distinction between being able to apply bioinformatics guided by someone who is an expert in a very narrow domain where you get to kind of see a very narrow perspective on what the application is, as opposed to understanding the broad spectrum and being able to decide where you would like to apply this on your own. And so as a result of this experience, we now have literally thousands and thousands of people that have completed such training and have found that this has provided them with a unique opportunity to see bioinformatics applicable directly to a topic that they might be interested in. And we have now a collection of different projects that we can point to that people have completed and successfully applied bioinformatics to these big data sets, like the Cancer Genome Atlas, like a variety of different projects on NCBI with publicly available data, as well as proprietary data that they have generated. So taking that as a starting point, I do want to mention that this is not some kind of a unique direction that only a few people are taking. We are talking about big data and biomedical research as both a unique business opportunity, an opportunity for entrepreneurs, people that understand the value of big data analysis to extract interesting insights from that, and specifically an application to biology or biological and biomedical data. And why is that so? I think that one of the important things is that the data is continuously growing, and it's growing in volume, in complexity. It is growing in diversity, diverse types of data. And a lot of companies that have seen this problem have focused on development of tools, have focused on effective ways to store data, manage data, process data, analyze it, maybe visualize it. And I think that that is definitely an important component of this problem. But I think what many have missed is that at the center are people. And these are diverse types of people. There are technicians, lab students, there are going to be clinicians, researchers, faculty. So it really represents a diverse set of users that have different backgrounds, different ages, different perspectives on the data. And all of them are important, because what we have found is that it is critical to address this bioinformatics or big data problem at all levels of data generation, data analysis, and decision making, because you have these people that stand at the generation side. They need to understand what kind of data needs to be generated and how to make data better descriptive for the different types of analyses that are going to be performed there. They need to understand how to eliminate batch effects, how to consider different types of data that represent the diversity of the experiments that they design. Then there are people that stand at the middle that have to integrate and analyze data to present the data to decision makers. And so for them, I think it's important to understand not only the technical part of the data analysis process, but also to understand where's the data coming from and how is it going to be utilized. And then finally, you have the decision makers, people that are going to be looking at the data, at the insights that they are provided with, and having to make critical decisions, decisions that might affect the treatment of complex diseases or even survival of a particular patient. They have to make decisions about drug safety and efficacy. And so I think for them, the reason why bioinformatics is an important part to understand is because they need to understand where did this data come from and what are the methods that have been used to analyze and extract the insights that they're relying on. So what we have focused on is bioinformatics training for people that want to understand biology as a data science. And so what we do is create biological examples that leverage data to explain how the data was generated, what are some of the statistical and mathematical properties of that data, how that data could be analyzed effectively, and provide tools that are logical and intuitive for people to use and regularly apply in research projects. And so this program that we are going to be starting is really focused on these kinds of topics. And so what I want to speak to you briefly about is how there is a growing demand for data literacy and skills in life sciences. Importantly, the industry is going to be looking at research projects that you can develop that provide you an opportunity to stand out. If you're planning your career in life sciences, in the industry, or in academia, the way to stand out is to really develop research projects that you can demonstrate your understanding and skills. We will go through some examples of projects and specific applications. We'll talk today about genomic data. We'll talk about how this program can help prepare you for a career, whether you're at the very start of thinking about where you want to apply these skills, or maybe you are already trained and you would like to maybe change your career, or maybe even take that career into a different direction. And then we want to talk about the outcomes for completion of this training. As Bipsa already mentioned, what are you going to gain as a result of participating in just this narrow program about genomics? Or we hope that you would consider joining the full program, which is going to span a much longer time. And also you will get introductions to mentors that are going to be very important for you to, as you build your network, as you continue to build your network in this field. So let me briefly describe this first part of this program, which is genomic data analysis. So this is based on a program that we conduct regularly. It's called OmicsLogic Genomics. And here we're going to be focused on specific applications of genomic data to the problems that are being solved by industry and academia, and how it is actually working. What is the data? Where does it come from? What do you do with it? So let's start with the very basics. What is the DNA code? As you know, the DNA code is a combination of nucleotides organized in a long sequence, about 3.2 billion base pairs. So what is a base pair? As you can see here on the screen, the base pair is a connection between two nucleotides. And so we measure typically the DNA in length of nucleotides or base pairs, and there's over 3.2 billion. Now, this 3.2 billion is organized in a specific way, but when we analyze this data, what are we looking for? One of the things that we're going to be looking for are DNA variation. And DNA variation means what are some of these example positions that we should consider as we think about how they affect phenotype? How do they affect the function, protein structure? What do they actually do? And the critical technology that allows us to ask questions about DNA variation is a technology called next generation sequencing. Now, next generation sequencing or second generation sequencing is a type of a sequencing technique that is high throughput. And what does it mean, high throughput? That means that DNA is largely broken out into small little short pieces, and then we have to piece those pieces back together into whatever region we are trying to analyze and start asking questions about variation that could explain what we are observing at the phenotypic level. So one of the things that you might think about are what are some projects where you could apply such analysis techniques? What could you actually address with some of these types of data? And so in this program, we're going to be covering a few different types of examples. There are going to be examples on drug metabolism. How does DNA variation affect our ability to process drug metabolism? So how do we metabolize these drugs, and what kinds of mutations might interfere with this metabolism? We'll talk about cancer and somatic mutations. Does anyone know what's the difference between germline and somatic mutations, by the way? Again, you can post your answers here in the chat. What are the differences between germline and somatic mutations? Okay, I'll give it a moment. Anyone can post their answers. Okay, so Deepak is saying germline can be inherited. Okay, excellent. And anyone else? So what are somatic mutations? All right, so let's germline during embryogenesis. Okay, yes. So germline are the ones that are passed from parents to their progeny, to their kids, and somatic are the ones that are acquired as a result of different processes in cell division, right? So after those cells have been, okay, excellent, Rapsan. Germline mutation in germ cell lines like sperm, somatic mutation in any other cells, exactly. Okay, and so somatic mutations are believed to be some of the primary causes of cancer, even though there are some germline mutations as well, right? Like BRCA1, BRCA2. And so we know now that in cancer cells, one of the things that we should be looking for are not just any somatic mutations, but somatic mutations in specific oncogenes, in genes that drive the development of cancer. So we're going to be talking about that. We are going to be talking about germline mutations in terms of inheritance of specific properties that have an effect on phenotype. For example, sickle cell anemia, which is an example of a project where you will be able to ask questions about whether a germline mutation is present or not, and also how does it affect the specific development of red blood cells. We'll talk about rare genetic diseases. So in genetic diseases, these are inherited diseases that typically manifest over time, and so you can either detect them at an early stage and do preventive therapy, or you could address them at a later stage, and there are things that could be done there as well. And then we'll also talk about mutations that are not in human or host cells, and they're actually in pathogens, so parasites, bacteria, fungi, as well as viruses. And so we'll talk about how these mutations could be detected, how they could be converted to amino acid changes or the language of proteins, and then how can we actually map them onto the 3D structures. And so the first thing that we want to start with as we consider this is the process of next generation sequencing. And so the first part of this program that I think everybody will have to kind of learn about and appreciate are the methods of next generation sequencing. And those include several different methods that have to do with data preparation, or how does the biological material get converted to this digital data that we're analyzing. And then the major second part of it is how do we actually analyze this data. Now, as Beepsa mentioned, a lot of what we will do together in sessions like this, but they really rely on these online asynchronous resources that you have access to and you will benefit from completing before every session. So in every session that we will talk about a particular topic, we will ask you questions about a resource that you have completed online in an asynchronous way. And when you have completed it, you will have several different important things, which include theoretical materials. They include practical exercises. Some of those exercises will be visual, and some of those exercises will be coding. So you will do a little bit of coding in R and Python to really understand what's behind some of the methods that we're doing. And then you will also gain access to projects. So Beepsa already mentioned several of the project resources that you have access to. But for those of you that have not completed the registration yet, I want to mention that these are the training materials, data analysis tools, curated project data sets, and interactive sessions for project-based learning of bioinformatics and data science that will be compiled together to provide you with a comprehensive, in-depth experience where you will be able to learn bioinformatics, apply it to important projects, and also showcase some of the projects that you have completed as a result of this program. So what our hope is for those of you that are joining this program is that you will be able to identify the connection between data and the application of that data analysis to a particular topic. Number two, you will gain some practical skills. You will be able to do this process from beginning to the end. You will be able to identify a data set, know what methods to apply to analyze such data, and then understand the biology that you're discovering as a result of your data analysis, as well as knowing how to explain, interpret, and present what you have done to an audience of people that are either just like you or actual experts in bioinformatics. And so to speak a little bit more about some of the examples, I think we have assembled a team of mentors and experts that will be able to provide you with excellent advice and feedback on all aspects of your project. And one of those mentors is Dr. Harpreet Kaur. She has in-depth experience in bioinformatics. She has a PhD in bioinformatics and has extensive experience in cancer genomics. So to speak a little bit about her background and how this program will help you understand genomics, I want to invite Dr. Harpreet Kaur and speak a little bit more about what she will be presenting in this program.
Thank you, Ilya. So, hello everyone. My name is Harpreet Kaur. I have a PhD in bioinformatics, specifically focused on liver cancer. In my current role, I'm curriculum developer and research consultant with Mind Biotech. Here, I'm assisting participants to learn about application of the machine learning in diverse fields, including infectious disease and cancer genomics. So recently, we have completed a research project entitled "Effect of Circulating AMA haplotype variant in Ghana on interaction with RON2 for merozoite invasion and vaccine development strategy," in collaboration with Dr. Anita Ghanshal's team at Noguchi Memorial Institute for Medical Research, Ghana, using NGS genomics data. We are looking forward to publish this work in high impact journal. So beside these original research projects, to provide hands-on training to make participants familiar with the approaches to design and complete research projects, We are continuously designing different research projects using publicly available data based on the studies that were published in high impact journals. Thus, we have a collection of research projects where you can learn how bioinformatics can be applied using publicly available genomics data to drive biological and clinical meaning in the diverse fields. Now, I would like to pass this session to Dr. Mohit Mazumdar, who will talk about complete program.
Hello, everyone. So my name is Mohit Mazumdar, and I have my PhD in computational biology, and I've been working on bioinformatics and bioinformatics projects for over 12 years now. So today we wanted to discuss and start this 4-month intensive research program. So the first phase of this program is Genomics for Biomedical Drug Discovery. So today is our first session, and I'm really happy that many of you have come up and joined this program and are now going to participate in this research journey with us. So in this process, we will also discuss the DNA sequencing and how analysis of whole genome and exome sequencing data can be used to find variants and study association between genotypes and phenotypes. So the next session is about coding. We will also be learning how we can start from simple coding lessons in R and Python in context to understand bioinformatics and application of those sequences. So we'll be learning about working with sequences. We'll be learning about how to load DNA sequences, look at RNA sequences, protein sequences, and that we will be doing through our coding platform using R and Python. So we'll be emailing you all of these content as many of you have registered, and we'll be getting the access and all the tutorials before the session so that you can also practice it on your own and work on it while we go and present it, so that you have more questions to ask, so that you have more insights into what we are talking. So after that, we will be learning about sequencing. So what is the history of NGS, overview of DNA structure and the DNA variations. We'll be discussing point mutations, deletions, duplications, frame shift types of genomic sequencing. What are the different types of genomic sequencing? So the targeted genomic sequencing, or the whole genome sequencing, or the whole exome sequencing. What are the advancements and what are the challenges? Also, we'll be learning about the application in the field of drug designing and discovery. So this will be on 30th of April. So we will have sessions after a gap of 1 day, so that you can review the material. And one of the aspects of OmicsLogic program that I wanted to mention, and I would want you to take a note of it, that we will also be sending you the recording of the sessions. So which means that if you are not getting time during the time that we are meeting regularly, you can also do these during the weekend when you have time. So you have to go through the videos, and we will be sharing you all of that using email and also through our Slack channel. So I think towards the end, Deepsha will be showing you the communication ways, the way we are communicating with you all the time and available and answering your questions through our Slack channel, which all of you have been invited to who have joined this program. So after that, next we will be doing the Genomic Hands-On, so introduction to genomic. So we will be learning about also what all other technologies that are there. So microarray, high throughput screening, qPCR. So what are the advantages, and about different kind of machines that are used, like Illumina, Nanopore, or PacBio. After that, it will be about the application. So biology of genetics, and how this is specialized in the sense how this is being applied to cancer research or precision oncology research. So we will be learning about how we can identify variants, and then how we can do visualization using integrative genomic viewer. So looking at the chromosome, and then looking at those genes, and then finding those exons and those mutations. And then it will be about projects. So how we can plan bioinformatics genomic analysis, how we can do literature review, and how we can start compiling a primary data set and then developing an analysis plan. So we will be covering that, and we will be sharing a project feedback form to you, so that any interest that you have in genomic data analysis, and if you have any questions that you would want to work on, you will be able to submit that and work with our team with that project proposal. So after this session, we will be talking about the application, in the sense what technologies could be used and how multiple sequence alignment of these large or viral genomes are giving us insights about the evolutionary analysis. So we will be also doing phylogenetic tree reconstruction of those genomic sequences, and then finding out about the mutations and the variants. So the germline and the somatic mutation that Ilya discussed, and then the practical assignment would be about the genomic variant analysis, and we will be also using external tools such as genome browser. So after that, after you have an understanding of how you can do genome analysis, genomic data analysis using the T-Bioinfo platform, then we will be discussing about how we can do this for visualization, and then we will be using filtering algorithms using the R code. So this will be a coding lesson, and again, we will have coding lessons along with the live sessions, so that you learn also these coding lessons through the platform. And we have a very easy to use interface. So we have all sort of students who are doing this analysis, coming from high school to postgraduate and faculty. And I think they have we have been able to make the platform suitable for all of them by incorporating the advanced research aspects as well as the basic part. So it starts from the basic, and you'll be able to learn how to do this for sequence and structural analysis. And then we will be discussing next that what we will be doing in the sense of the project, and how we can understand or correlate with the function. So this will be about the sessions. Sorry, this is a wrong date. I think we'll have to correct that. So 17th May, the program will be finishing this first part, and then we will be moving to the second data type, which is equally important and is in more use in biomedical research, is the transcriptomics data. So in the second phase of the program, we will be learning about the second data type, which is to understand the application of gene expression in biomedical research, and what different kind of innovations and research is being going on. So in this detailed program about transcriptomics, we have been doing this program for quite a while now, and have several projects that students have been able to finish and come up with. So a couple of projects that you saw from Urja and Dilara, they kind of discuss different kind of data types. So one of the major data type that we are here kind of going through is the transcriptomics data type. So we'll start from introduction to transcriptomics, then how we can process data, transcriptomics gene expression data using different algorithms that could be used, that flows from one kind of analysis to another. So we will be learning about pre-processing. We will be learning about how we can map it to the genome, and then generate a table of expression, and then what kind of statistics and how we can gain insight using machine learning algorithms, and using different kind of regression and statistical analysis, and then R and Python into it, and then we can come up with a biological interpretation. So it's a very it's a kind of scientific program that is being appreciated by the entire scientific community. We have more than 5,000 user feedbacks that we have received about this program, and I think it will be much appreciated once you go through this. So in this, towards the end, we will be talking about the components and how all of this different type of learning that you're getting could help in a translational bioinformatics project. So a pharma project, for example, for drug discovery. So one of the major challenge when it comes to clinical data is also the noise. So there are many available observations that are used by clinicians without a meaningful connection to the actual data state. So this methods that we use for using machine learning is to be able to do this analysis and can separate meaningful measurements from those that behave in a random way. So we have to identify those. So our team actually worked to evaluate this measurement system to a disease to help select responders to a combination of treatment. So this symptoms associated with disease state were separated between those that are relevant and those that are not relevant to prescribe drugs. So this is an example of precision medicine, and also how we can find out those patients or those biomarkers to help in proper diagnostics. So using this technique, drug impacting the patient condition could be identified. So plays an important role in pharmaceutical industry and also for drug discovery and designing. So that's one of the aspects that I wanted to tell you about. Then next we will be learning about the machine learning. So this is the phase 3. So all of you know that data science has potential to make an impact in all kind of operational pipelines in a company. So it could be a pharma, it could be business, it could be academia. So so far, especially in pharma, the data science has already made a huge impact. So it's being used now for clinical trials. It's being used for drug discovery. So we will be learning about all of that in the second phase. So this will be an intensive data science program where we will be having an introduction to data science and algorithms. So we'll be discussing about the mathematical algorithms. So Dr. Sujit here, who's a data scientist in one of the leading company, is going to take some of the sessions here, and we'll discuss the application of data science. And then we will also, our OmicsLogic team will introduce you to the machine learning aspects of NGS data and how we can use NGS data for different kind of data mining, and how we can use R and Python to come up with better visualization and understanding of the data that we are looking at. So we'll be learning about PCA, different type of unsupervised machine learning methods, such as clustering. And then we will be also learning next about the predictive analysis. So that would be about random forest, that would be about support vector machines, and then how it could be used in validation. So what are the different validation methods that are there? So 5-fold cross validation, and then how machine learning is used in production. So what are the application? And then there would be a project based session which is scheduled on July 12th. At the same time, we will also be starting a parallel program that will run on Cheminformatics. So machine learning and Cheminformatics will go on, and you will be learning about the 3-dimensional data. So in this entire process, what we will be covering is that about the omics technology. So to all of you maybe who are not much aware of the omics technology, and maybe I should pause here and maybe ask you that how many of you here who have joined us have an experience of working or have some understanding of the application of omics data in research projects. So maybe you can write 1 for having an experience, or 2 for not having an experience. So please let me know. Okay. Hi, Laura. So yes, I know that you have experience. So and I see a lot of students have not exposed to this technology yet. So maybe I can explain this in a few sentences. So omics technologies are defined as high throughput biochemical assays that measure comprehensively and simultaneously molecules of same type from one biological sample. So for example, genomics is to profile DNA. Transcriptomics is to measure transcripts. Proteomics and metabolomics are to quantify proteins and metabolites, and so on. So this omics notion refers to the fact that all or nearly all instances of the targeted molecular spaces are measured in the assay. So it's a very powerful technology if we can give them, like if we can have an integrative understanding and how this data points are connected. So therefore, like an holistic view of the biological system, right? So initially, omics experiments are used to concentrate on one type of assay, for example, transcriptomics, and provide single omics data. But now the technology has progressed to multi-omics by integrating multi-omics data. And we also have the platform that integrates this multi-omics data to be able to come up with those kind of insights. Anyways, this would be one of the aspect. And now the next, as I was telling you, that we'll be also running at the same time Informatics for Biomedical Drug Discovery, and have an amazing team with us here today also, who would be helping you going through this training. So this training will involve introduction to Informatics, which would be structure, chemical interactions, and databases. And then you will be learning about virtual screening. So how this can be done in a virtual environment where you have a defined protein structure, and then you have a defined active site, and then how you can screen number of molecules and maybe make sense of that data, that how this is useful. So the next step after that is to do hit identification. So hit identification is that once you have like a list of good binders, and you kind of think that they could be good molecules to go on with that. So then based on those, you can again do chemical similarity search. So that would be one of the aspect. Another aspect is to do QSAR, and based on the understanding of the binding site. So if you have a model, or if you have a crystal structure, so all of that would be important. So then the aspect of finding that great pose. So that great pose is in the sense of the structure. So like we do click pictures with great pose, so we kind of try to find great pose in docking. So when a molecule is being docked, and we would want to find a better energy state of that molecule at a bound form, so that we can maybe hypothesize that it could be a good drug molecule. So from hit to lead modification, and then chemical compound modification, which is rational drug designing, then admit predictions. And also we have Dr. Sujit, so who is now working on image classification, so that he's calling it like computer vision. So that would be also a part of this one of the sessions, and examples of successful and failed drug development, and then data science and AI and clinical trials. So this would be like a combination of chemoinformatics and data science, and application of chemoinformatics in data science. So this is a very unique opportunity for all of you to learn from this this part of the program. So yeah, so I think we kind of discussed that we will be going through several of these examples. So Curaxin is another example. So a cancer drug that is in clinical trials. So yeah, we'll be discussing those in detail during the session. And then we have as you learned from Dr. Harpreet about malaria bioinformatics. So we are working on several projects. So you'll also have opportunity to do some of the research if you're interested in malaria. So overall, I think this entire program can help you learn about different kind of diseases. So biomedical application, after learning about what are different data types can contribute, and how we can connect those data from a target discovery to a molecule for the problem. So the entire schema of drug discovery. So as I said, this summarized my presentation, and this is what we have planned for you for this summer. And if you have any questions, please feel free to ask us. And now I will pass it on to Beepsha to talk more about how you can make the most out of this opportunity and what are all resources that we have and how can you register for the program. Thank you. And if you have any questions, please put it in the chat box and I'll be able to help you there and answer those.
Thank you so much, Dr. Mohit. So I want to again take everybody back once more to the website so that we can go through the registration process. And also I want to introduce you to our Slack channel so that you can understand how you are going to be using that. So I'm sure by now, most of you are already there on the platform. So if you are not yet creating an account and setting up an account, please go ahead and do it so that you can gain access to all of the courses and start going through them. So please feel free to go through these courses. And one of the best aspects of going through some of the courses like R and Python would be this, that you do not have to install R and Python in your computer. We have provided code blocks right in the web browser. So you are going to be able to use the web browser to go ahead and do those lessons. And again, if you want to, you can go ahead and download the applications or the software and do it in your computer as well. So you have both the options. So please feel free to go through some of the courses that I have mentioned earlier. The one is Introduction to Bioinformatics. And on page 3, you would be finding the course on genomics, which is the topic of this entire 1-month program. So please feel free to go ahead and start with this. All participants who are already registered for the program will receive an email from marketing@pine.bio where you would have your individual server password. This access will come with 5 terabytes of space in the cloud. So when you run your pipelines, you'll have 5 TB of space. And of course, in the upcoming sessions, we are going to be telling you how to use that. And you will be learning a lot more about how these pipelines work. So please make sure that you have all of these access details. And if you have any questions regarding any of these resources or you're not able to set up your account or face an issue, please inform us before the session so that you're able to follow the mentors during the session as we go forward. And let me also show everybody the Slack channel. So this is our Slack channel on biomedical data science and drug discovery. We will be posting here, for example, I had posted here the Zoom link to join today's session, and we also had many members join here. Similarly, we'll also be posting the videos of the session here. We'll have discussions, so please feel free to use the resources. Okay, Vijay has a very good question. How long can you use the resources that are mentioned? So if you are already signed up for the full 4 months program, you would be having access for the entire 4-month period. And in that, if you have already gone ahead and paid for an advanced license, you would have 30 days more to go ahead and access the resources. So it becomes a 5-month access to resources. Now, if you are only registered for one part of the program, say you're only registered for genomics, so you have access for the next 30 days. And after May 18th, if you have an advanced license, you can again access it for 30 more days. So those are some of the licenses I will be showing you shortly. So for the Slack channel, this channel is by invite only for any of the participants who are already in the program. So we will be emailing you the details of the Slack channel only via email. So only registered participants will have access to the Slack channel and direct access to mentors for them to be able to interact with our team as well as to talk and post any of their questions and also interact with each other. And Akanksha, yes, sure. This session is being recorded, as will all of the upcoming sessions. So you will be receiving links to all of these recorded sessions. So we'll post those links here as well. And we'll also email you those links so that they're there in your email inbox. So you do not have to worry about that. Do we have any other questions related to the resources that we just discussed or whatever we have covered today? What we have covered, like for Dr. Harpreet, Dr. Mohit, or Elias part. So please post those questions right now in the chat box, or you can also type in your question out here and we will be getting back to you. And now for any of you who have not yet joined this program, so we still have the registrations open for the full session. So if you are joining the full program, all you have to do is go ahead and click on get started, and you will be given these options. Now, as I was mentioning, if someone has chosen the advanced license, they'll have an additional 30 days. So this gives you ample time to work on your project. So what I wanted to tell you is this, that if you are working on a project, then we have seen that 3 months is typically the time that it takes for some of the projects that Dr. Mohit and Ilya mentioned, as well as Dr. Harpreet showed you during her presentation. So typically it takes about 3 months to have come up and refine those projects. So you have a period of 5 months, and we'll soon be talking about how to develop those projects. So it's really beneficial if you have some more time to work on those projects and work with the mentors on getting the feedback. And here I have a question. Will there be a disconnect if we enroll only for phase 1 and 3? Will I be able to follow phase 3 without having to attend phase 2? So these programs are designed in a very step-by-step manner where we are introducing you to certain concepts. However, we are giving it open to you for all the participants to choose as they would want to, because many people may not be interested in genomics, but they may be interested more in transcriptomics or in chemoinformatics. So you can, but we'll start each of these sessions, of course, with a more introductory sessions at the start of each of these programs, and then develop those concepts. However, anyone who has been going through the training for, say, the full 4 months period would, of course, have access to more resources and more time with the mentors, as well as more sessions that we would be doing. So there may be a disconnect depending on what you are working on. However, if you're just choosing those individual topics, it is fine, because we will be covering genomics starting from today, then transcriptomics, then data science, and lastly, chemoinformatics. And I also wanted to work everyone through the genomics registration. So if you're just interested in registering for the first half of the program, which is starting today, again, you have the option here, start from the beginner, and we also have an option for taking 55% off for this entire training. So make sure that you're putting in the coupon code that has been emailed to you in this and clicking on apply. Wait for a second so that it shows that the coupon has been accepted, and then you can go ahead and put in your card details. And if you're paying any of the mentors, please feel free to reach out to them, either Faba Academy or Pro Biotechnica or Pharma Toppers. And you can also reach out to us, marketing@pine.bio, again, for any questions in regards to the resources and the training, as well as registration. And we will be happy to help you out.